Hey guys, welcome back. Now let's move on to renal physiology. This is where the bulk of the material will be for renal because so much is known about the complex and really amazing physiology of the kidney. Let's first start off with the big picture, looking at the body's fluid compartments. Now recall that the total body water is split into several compartments. An easy way to remember the ratios is the 60-40-20 rule. The entire body is made up of 60% water. So that's where the 60 comes from. And 40% of the body is non-water mass. But this is not where we get the 40% in the 60-40-20 rule. Because the 60-40-20 rule is only talking about how water is spread out throughout the body. Now if we go back to the total body water, 2/3 of the total body water is intracellular fluid. And this is where the 40% comes from. Because in other words, the intracellular fluid makes up 40% of the total body weight, which is also 2/3 of the total body water. The extracellular fluid, on the other hand, makes up 20% of the body's entire weight, which is also 1/3 of the total body water. So the 60-40-20 rule means that 60% of the total body weight is water and 40% of the body weight is intracellular fluid and 20% of the total body weight is extracellular water. Now, the volume of the extracellular fluid can be measured by inulin. And we'll see some of the benefits of that later. But inulin can move in and out of the vasculature because it's not taken up by cells. So therefore, the inulin remains in the extracellular fluid. And the extracellular fluid can be further divided into two compartments. The plasma volume, which makes up 1/4 of the extracellular volume, and interstitial volume, which makes up 3/4 of the extracellular volume. Now it's intuitive that interstitial volume is going to be bigger because that's the volume around all of our cells, whereas plasma volume is only the volume of fluid inside the vascular system. So plasma volume is measured by radiolabeled albumin because the albumin protein is too large to pass through the fenestrations of the capillaries. Now, before we get into the differences between these compartments, let's walk through a sample problem. Now, how is the body water of a 70-kilogram man going to be divided up? If you weigh 70 kilograms, then how much of that will be water? 60% will be water, which is equal to 42 liters. Then 2/3 of that is going to be intracellular, which is equal to 28 liters of ICF. The remaining 1/3, which is equal to 14 liters, is going to be ECF. And of that ECF, about 1/4 is going to be what? Plasma volume. That's right. So that gives us 3.5 liters of plasma volume. And the other 3/4, which is equal to 10.5 liters, is going to be interstitial volume. Now, these kinds of calculations are important, especially for pharmacology, where you have to calculate the dosage based on a drug's volume of distribution. So if you need to, go ahead and pause the video and see if you can follow my calculations. Now let's talk about the differences between these compartments. The extracellular fluid, or ECF, has a very different composition than the intracellular fluid, or ICF. The major cation in the ICF is potassium, while in the ECF it's mainly sodium. As you remember, this same concept is taken advantage of by the nervous system to facilitate action potentials. Sodium, which is a major extracellular cation, enters the cell, and potassium, the major intracellular cation, exits the cell. So the ECF and ICF also have different anions. In the ICF, it's mostly protein and organic phosphates. Remember that the majority of the body's proteins and ATP are inside the cells. In the ECF, it's chloride and bicarbonate. Remember that bicarbonate is used as an extracellular buffer system as well as a means to transport CO2, and chloride is used to balance the charge shifts as bicarbonate moves across the red blood cell membrane. And do you remember what this is called? This is called the chloride shift. And when we look at the osmolarity of both compartments, it's about the same at 290 milliosmoles, which means there's no net movement of water in or out of the cells at rest. Let's try another practice problem. You infuse 1 gram of radiolabeled albumin into a healthy 70 kilogram volunteer. I hope he's still healthy afterwards. After equilibrium has been reached, the plasma concentration of radiolabeled albumin is 25 mg per deciliter. What is your volunteer's plasma volume? The answer is 4 liters. Now let's walk through how we got that together. If we infused 1 gram, or in other words, 1,000 milligrams of albumin, and the concentration in the plasma ended up being 25 milligrams per deciliter, and we want to figure out what is the volume, we just divide 1,000 divided by 25. Now the mgs will cancel out, and we'll get 40 deciliters, which is equal to 4 liters. And that's roughly equal to 3.5 liters that we would have estimated based upon our knowledge of the fluid compartments. So for your test, it's important to remember that various substances can serve as surrogates for different fluid compartments. For example, inulin will often be used on the test as a reference for which compartment? The ECF, and this is worth remembering. It's also useful to have a general idea of the volume of each compartment for an average human being, because if your calculations come out to be really messed up, like 30-liter plasma volume or something, then you can go back and check what you did wrong. Now let's move on to the actual functions of the kidney, starting with filtration. The glomerulus is a very delicate structure that acts as a filter for plasma. The way that the glomerulus achieves filtration is a variation on a common theme in human anatomy. If you remember the blood-brain barrier, it is comprised of 3 layers: an endothelial layer, a basement membrane, and astrocytic end processes. The glomerulus uses the same concept. It has a fenestrated capillary endothelium on the inside. There's an endothelium, and that acts as a size barrier. Then it has a fused basement membrane that acts as a charge barrier because it has heparan sulfate in it, which repels negatively charged molecules. And instead of astrocytes for the third barrier, it uses podocytes that you can see here on the outside. The podocytes are the third barrier. And these barriers are especially important to keep protein, which tends to be large and negatively charged, inside the blood. If this barrier is lost for some reason, the glomerulus will leak protein, leading to decreased oncotic pressure and edema, as is the case in nephrotic syndrome, which we'll study in the future. Normally, only small solutes are freely filtered through the glomerulus. What are some of these solutes that are freely filtered, you might ask? There are things like amino acids, glucose, and electrolytes. Hey, wait a minute. Aren't these solutes that we want to keep in the body? Why, yes they are. But the problem is that these solutes are the same size as the waste products which the kidney is trying to clear from the body, like urea and creatinine. So the glomerulus has no way to separately filter these solutes, so it filters all of them and then reabsorbs the ones the body wants to keep, like amino acids, glucose, and electrolytes. Now let's move on to some equations that are important for renal physiology. These equations are important for determining whether the kidneys are functioning properly as far as retaining certain things and letting other things filter through. Once you understand the concepts behind the equations themselves will become intuitive. First, we have renal clearance, a concept that you need to know, especially for pharmacology. Now, clearance describes the volume of plasma in milliliters that is cleared of a certain substance, in this equation X, over time in minutes. Let's say that after infusing substance X into a patient's blood, we come back an hour later to find that the plasma concentration, in this case px, is very high. Now what does this mean? This would mean that the clearance of substance X is pretty low, which makes sense with the equation. If px goes up, cx goes down. If, on the other hand, we've found out that the urine concentration of substance X was very high an hour after infusion, then what would this mean? This would mean that the clearance of substance X is pretty high. Again, it makes sense with the equation. It's also intuitive that with higher urine flow rates, in this case V for urine flow rate, the clearance of any substance will be higher, because more urine can only be produced if more plasma is filtered through the glomerulus, assuming that there aren't any other changes occurring in the downstream nephron. Measuring the clearance of a substance and comparing it to the glomerular filtration rate, the GFR, can tell us how the nephron processes the substance. Remember that the GFR is the amount of plasma that is filtered through the glomerulus per minute. So, if the clearance of a substance is less than the GFR, then what is the nephron doing to the substance after it's filtered? It's reabsorbing it, because there's no way a freely filtered solute can have a clearance less than the GFR unless it's being reabsorbed. And what if the clearance of the substance is greater than the GFR? Then that means the nephron is secreting the substance, because that's the only mechanism after filtration by which a substance can enter the filtrate. So, lastly, if the clearance and the GFR are equal, then there is no net secretion or reabsorption. As I said previously, the GFR is the amount of plasma that is filtered through the glomerulus per minute. This can be measured using the inulin clearance. So here we see inulin again. And remember that inulin is not taken up by any cells, but it's also freely filtered and is neither reabsorbed nor secreted. So that's why it's used to measure the GFR. Now in clinical practice, everyone uses creatinine clearance because it's easier to measure, but keep in mind that it is a slight overestimate due to creatinine secretion via the tubules. Now GFR is used as a proxy to measure kidney function. It represents the balance between the Starling forces in the glomerular capillaries, which are the hydrostatic pressure trying to push fluid out, and the oncotic pressure trying to keep fluid inside the capillaries. In the bottom equation that you see here on the screen, Kf is the filtration coefficient. So what does that mean? The filtration coefficient is basically dependent upon the resistance to fluid flow through the filtration barrier of the glomerulus. A higher Kf means less resistance, which would increase GFR. So what is something that could lower Kf? One example is damage and scarring to the glomerulus. So what two variables in this equation, if we increase them, would increase GFR? The answer is PGC and pi BS. PGC is the hydrostatic pressure inside the glomerulus, and pi BS is the oncotic pressure inside Bowman space, the area just outside the glomerular capillaries. And of course, increasing PBS, the hydrostatic pressure inside Bowman space, or pi GC, the oncotic pressure inside the glomerular capillaries, would decrease GFR. So let's do an example calculation to test our knowledge of clearance and GFR. Calculate GFR in mils per minute for a patient using the following values of inulin. Urine concentration equals 100 mg per deciliter, urine flow rate equals 50 mils per hour, and plasma concentration equals 60 mg per deciliter. The answer is 1.4 mils per minute. Now let's see how we got that. Using the formula where GFR is equal to inulin clearance, then that means that GFR is equal to the urine inulin, which was equal to 100 mg per deciliter, times the urine flow rate, which was 50 mils per hour, divided by the plasma sodium concentration, which was 60 milligrams per deciliter. And when we do that calculation, we get 83.3 mils per hour. And dividing that by 60, we get 1.4 mils per minute. Let's try another question. If the same patient in the previous question has the following concentrations for sodium, urine sodium equals 70 milliequivalents, urine flow rate equals 50 mils per hour, and plasma sodium equals 140 milliequivalents, does the patient have net sodium reabsorption or secretion? The answer is net reabsorption of sodium. So let's work out how we got that. Again, using the formula for clearance, and this time the clearance of sodium, we get 70 milliequivalents of urine sodium concentration times 50 mils per hour urine flow rate divided by 140 milliequivalents plasma sodium concentration. When we work that out, we get 25 mils per hour. And dividing that by 60, we get 0.42 mils per minute. And now we have to compare this to the GFR, which was 1.4 mils per minute. So we see that the GFR is higher. It's greater than 0.42 mils per minute, which is the sodium clearance. So that means because the GFR is higher than the clearance, there is net reabsorption of sodium. So be comfortable with these equations because the USMLE will test your knowledge of them both conceptually and mathematically.